Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Welcome to the next session in the Health and Wellbeing track. It gives me great pleasure to introduce you to our next speaker. Jennifer Listgarten is a researcher at Microsoft Research, and she'll be talking about hidden confounders in genetic analyses.
Simon. Okay. So just a brief outline, I'll tell you the importance of genetic studies, which I don't know what the background of people in this room is, but I'll do that, and then just go over some basic terminology, which may at this point be redundant given some of the other talks. And then I'm going to focus in on the problem of genome-wide association studies from a statistical methodological point of view, not focusing on any particular disease. And then I'm going to finish off by telling you about some recent work of ours that was published specifically on eQTL analyses. And if you don't know what that means, I'll get to that during the talk. So I would say that the era of personalized medicine has arrived. And so what does that mean? It means using the genetics of people to tailor anything in medicine, really. So, for example, 10% of people don't respond to codeine. So if you know that, you don't want to give them codeine. In the area of HIV, there's a drug called abacavir, which you can react to very, very badly. It's highly toxic. And if you know you have a particular genotype, then you know not to give that person that drug. And there are many drugs that you can tailor the dosage to based on a person's genotype. Another area that was briefly mentioned by a previous speaker is disease subclassification. Sometimes there are diseases that are thought to sort of be one disease, but when you look more deeply, after you've generated a lot of data, you can gain insight and see that there are actually subclasses of this disease, and that helps to tailor what treatment you would give to those patients once you recognize that they're actually distinct subgroups. And there's also the issue of risk identification. So if you know that particular genetic markers make someone more susceptible to, say, colorectal cancer, then you're going to want to track those people more carefully than other people to catch the disease earlier rather than later. And lastly, a lot of these studies are done just to gain basic biological insight into pathways and mechanisms of what's going on in the body. And that can translate also into drug discovery by finding out which mechanisms or pathways are involved in a particular disease. So again, a lot of these terms have already been used today, but I'll just go over them once again to make sure we're all on the same page. So this here is, what's drawn is the so-called central dogma of molecular biology, which just shows you the flow of information. So starting with the DNA on the top level, and where, of course, you have genetic variants, and I'm going to talk in particular about SNPs, single nucleotide polymorphisms, where you might have an A and I might have a C, and that might explain the difference between why you're susceptible to some disease and I'm not, for example. And then I'm going to talk about microarray expression data, which measures RNA levels, which you can think of as a proxy to protein, although they're not directly correlated by any means. But as it's highly informative, and so there's a lot of work done with gene expression data. And lastly, there's often a phenotype of interest, so for example, whether someone has asthma or not. And so as you've already heard from previous speakers today, genome-wide association studies, when you basically scan through many, many markers in the DNA, in this case I'm talking about single nucleotide polymorphism markers, and just one at a time, and asking if each one in turn is correlated with a particular phenotype of interest, like asthma or ALS. And I'm also going to, well, actually I'm not going to talk about this one, but it's relevant to eQTL analysis. So differential expression is another type of question you can ask, where you scan through all of the genes, all 20,000 genes, and you ask each one in turn, is that associated with my phenotype of interest? And what I am going to focus in on at the end of the talk is eQTL analysis, where you say, for each marker in my DNA, which might be 500,000, is each one associated with any of my genes? And there could be, say, 20,000 of those. And so that's a very large hypothesis space to explore. So what are some of the statistical challenges in this area? Again, as you've heard today, that you have a massive scale of multiple hypothesis testing, basically. The more things you test, the more likely something's going to look significant by chance alone, and you need to be able to correct for that. And one way, as we've heard, is a Bonferroni correction. But I would argue that increasingly people are going to other measures that make a lot more sense, in my opinion, such as the false discovery rate, which is just a different way of looking at how what you consider to be the so-called error rate of your statistical analysis. I'm not going to go into that for this talk. And the other issue is data heterogeneity and the problems that arise from that. So as you start amassing larger and larger data sets, you end up with people with different racial backgrounds, or you may have some sort of people who are more related because you've collected people in a family, and essentially you've violated the principle of a lot of statistical analyses which assume that people are identically distributed. That is, they're all sort of drawn from some homogeneous population, and that can get you into a lot of trouble. So you can flip around some of those challenges and actually sort of turn them on their head and make use of them. And so in particular, when you have this massive scale of hypothesis testing, it means that if you test, say, 500,000 SNPs, you get 500,000 p-values. And there are ways to look at those p-values that can give you insight as to whether your statistical analysis is correct or not. And I'll go into more details about that in a second. Basically, under certain conditions, you can assume that the distribution of those p-values should look a particular way. And if they don't, then you know that you have some problem in your statistical analysis. And the other issue is that I mentioned there may be different racial structure or different degrees of relatedness of people in the data. And it turns out that when you have lots of data, that you can actually infer these things from the data itself and actually correct for them. And so between these two things here, we can actually do really well even when we have many, many hypotheses to test and lots of data. So I'm just going to sort of go into more detail now with genome-wide association studies and tell you how you can see these problems and how you can correct for them. And then that's going to lead me into the work about eQTL analysis. So again, in a genome-wide association study, you basically start off with a large set of people that either they are case control would be one example, they have the disease or they don't, and then the desired output is a list of genetic markers associated with that disease. And so, again, I've already alluded to this. There's often problems of hidden structure. And again, I'm just repeating myself here, but just so that you understand, the problem is that when you throw together people of different ethnic backgrounds or there's family structure in the data, you end up with a confounding signal that dramatically changes the results. And I'll give you an example of that. And you need to correct for it. And so the end effect in a statistical analysis is you basically get many, many spurious correlations and a loss of power to detect the true signal of interest. So just to give you a bit of intuition as to how that can happen, this is an example from Nature Reviews Genetics where they've shown each circle here is a person, and it's separated by case and control. And it's also separated by population 1 and population 2. And so what you see is that population 1 has more A's than T's, and population 2 has more T's than A's. And so when you just split it down and say, is anything different between case and control, just because the proportion of population 2 is more in the controls than in 1, it looks like they have more T's than the cases, or conversely, that the cases have more A's than T's. And you would say, well, the A must be associated with being a case and is therefore associated with disease. And while that could be true, there are much better explanations typically for why you see that, and that is because you have this difference in alleles between the two populations. So basically, the problem is that you have this heterogeneous data set, and the proportion of population 1 and population 2 is different between your case and control. So what you're going to pick out with a statistical analysis is any position that just is different according to race rather than disease. And there are many, many such hits, far more than you would expect to see correlated with the disease, in fact. And so often people think that you can fix statistical problems by increasing the data set size. If you have more data, then things become cleaner and you increase the signal to noise ratio. But in fact, with this problem, what happens is you can amplify it. So the more people you have, the more power you have to uncover these population differences, and so the problem doesn't go away. So let me walk you through this figure here. This figure is meant to show you how you can use this large scale of hypothesis testing to see when you have a problem in your statistical analysis. So like I said, you scan through essentially, say, 500,000 SNPs, and you get 500,000 p-values, one for each SNP. Is this SNP associated with my disease, yes or no? And you have this number between 0 and 1. And in general, for a genome-wide association study, you would expect of those 5 million SNPs that very, very few of them would be associated with the disease. And if none of them were associated with the disease, then you know theoretically what the p-value distribution should look like. It should be a uniform distribution on 0, 1. That's the definition of a p-value. And if you expect a handful of things to actually be associated with your disease, it's not really going to change that distribution. If you think 100 out of things at most, I think, might be associated with, say, diabetes or ALS, out of 500,000, you're still going to expect to see a uniform p-value distribution. And so to the extent that you don't see that distribution, you know something has gone very wrong. And so that's one of the main diagnostic techniques in this area for showing that you've properly analyzed your data. And so this is a figure that shows that what it is a quantile-quantile plot. And if you're not familiar with those, what you want to see basically, well, I'll tell you, on the bottom are basically the expected log p-values from this theoretical uniform distribution. And on this y-axis, the vertical axis, are the actual observed ones. And the implication of everything I've just said is when you do an analysis and you plot a curve for your experiment, you want it to track the diagonal and within these error bars. And to the extent that it doesn't, you know that you've got a serious amount of what's called inflation, that is spurious signal in your data. And so this is actually not made up data. This is a real data set, and this is what happens if you do a naive statistical analysis. Looks like you have tons and tons of signal when, in fact, on this data set, when it's analyzed properly, you have very, very few. And in contrast, this is a model that corrects for that hidden structure, and you can see it's actually within the error bars. And in fact, on this data set, there is no real signal that we could pick up. So now the other thing I mentioned before was that you could use the large scale of the data to basically detect some of this confounding structure and correct for it. And so I'm going to tell you momentarily about a few methods that are in the literature right now that people commonly use. And the central theme is that they all, as a starting point, use what I call as a genetic similarity matrix. And so this is 3 examples here. Basically, you can think of every entry as the ith, jth, 2 people in the data set and how similar they are. And so in a data set that has no population structure, for example, there's nobody related to each other and they're all Caucasian, all you see is that on the diagonal, people are similar to themselves, but they're no more or less similar to anyone else. And this is an example where you have a lot of related families in the data set, which obviously looks quite different. And here is an example where you have 3 ethnicities in the data set. And so as soon as your similarity matrix looks different from this, you know you need to do some sort of correction. Although I wouldn't rely on the visuals, you can tell this statistically. And so the 2 approaches that make use of this kind of genetic similarity matrix are principal components analysis-based approaches. The famous one would be Eigenstrat, and linear mixed models, which is actually where my work has been. So I'll just give you an intuitive overview of how these methods work. So the naive method to analyze to do a genome-wide association study, one naive way would be to just use linear regression, where you have, you say, blood pressure level here, and you want to know which SNPs affect blood pressure level. And so this would be the value of the SNP for each person. This would be their blood pressure level, and this would be some weight in your regression model that says how important is the SNP for blood pressure. And you would run this model with the SNP and without, and you would use some sort of test which would give you a p-value for that SNP, and then you would repeat this for all 500,000 SNPs, and then you could look at your distribution. And in fact, that's what I did on this data set here, and you can see that it's terrible. You have a huge amount of inflation. So one of the other methods I mentioned was this Eigenstrat, or principal components-based approach. And what you can think of here is that you take this big matrix of similarity, and effectively you do some computation that looks for major axes of variation in this high-dimensional SNP space. And so the intuition is you can that I think they actually call them ancestry directions in the Eigenstraat paper. But basically, you get some directions in this very high-dimensional SNP space that are informative about ancestry, and you add them as covariates into your linear regression model. And when you do that, it actually works quite well for a lot of data sets, but there are also many data sets for which it doesn't work very well. And I'll show you an example of that in a second. But just to give you a sense of how richly informative this genetic matrix, similarity matrix is, if you do principal components analysis on that matrix, what it gives you is a set of directions. And now for each person, you can say how much of each direction do they have, and now you get two numbers for each person, and you can plot those two numbers. And if you do that, what's shown here is that you can essentially reproduce the map of Europe. On top is annotated in color where they know where people have come from. And each point here is a two-dimensional point which arose simply from doing principal components analysis on this matrix. And so I think that's a pretty cute figure just to show you that you can get a lot of information out of these matrices. And so it works well to capture broad race-based structure, but it's actually quite sensitive to outliers, and it can't capture fine-grained structure such as family-based structure. And so the third approach that I'm going to mention now, and the one that is, again, central to my work, is a linear mixed model approach. And this is just a sort of intuitive overview of it. What you think of now is instead of sort of reducing this matrix to a set of directions, as PCA does, you use the entire matrix, and you basically can sample hidden regression coefficients that correspond to ancestry, and then you integrate them out in a regression. If you're not familiar with these, I don't expect this to really give you a tremendous amount of insight. But basically, it's using this matrix in a more informative way and throwing less of it out. And this kind of model, we've found, works a lot better. So it captures multiple levels of similarity. You can mix in a variety of races as well as family relatedness. And typically, in my experience, this seems to work across the board very, very well. It's not sensitive to outliers because it's not just looking for sort of very strong directions of variation, but it's using the whole matrix. But it is pretty computationally expensive. So here's that same data set I've been showing you, where if you naively apply no correction, this is the black line you get. If you apply a PCA-based approach like Eigenstraat, you can see that it corrects it to a large degree, but it's still far outside the error bars. And in blue, you'll see the mixed model approach, which is exactly what we would like to see. And so this gives you a lot of evidence to suggest that for this case, you really would like to use a mixed model and that other models won't do very well. What time did I start at? 3:35. Okay. Okay, so these mixed models are applicable to genome-wide association studies and differential expression and eQTL analysis. Ooh, some of my slides are completely missing. Wow. I know why. That's the wrong talk. It's not the right PowerPoint. Let's see. All right, okay, now I'm back on track. Right, that's where I was. And so, right, these can be used for all of these mixed models can be used for all three of these, and I'm going to focus in now just very quickly on eQTL analysis, which we recently published in PNAS, and this is joint work, I should say, with David Heckerman and Carl Cady at Microsoft Research and Eric Schatt from Pacific Biosciences. So the key insight here is that you can think of an eQTL analysis. You remember I said eQTL analysis was looking for associations between SNPs and between gene expression. And I've told you about this whole area of literature that tries to address problems of confounding for genome-wide association analysis. What I haven't told you about is there's an analogous set of literature that tries to correct for confounding in differential expression analysis. And so what you need to do when you have eQTL analysis is realize that you actually have both of these problems here, because every time you say, is this SNP, are each of my SNPs associated with a gene, that's really a genome-wide association study. On the other hand, every time you say, are any of my genes differentially expressed for this SNP, that's sort of like a differential expression study. And so it turns out you get statistical confounders from both of these areas, and you need to address both of them. And so that's what's novel in this work, is addressing them jointly and the way in which we did so. So it's actually, given what I've already told you, superficially it's quite simple. I mentioned that there was this SNP space confounder coefficient that gets added to the regression and then integrated out. And that was based on a similarity matrix here, which actually I realized I omitted to tell you how this is created. This is basically you just take all the SNPs that every two people have and you say something like how similar you count the number of times that they share the same SNP, more or less. And that's how this one is created. So now we're trying to correct for gene expression confounder. And so what you want to do is say how similar are every two people in gene expression space. And again, you can use their gene expression data and basically compare the similarity of their gene expression profiles and put it in this matrix. Although I'm glossing over a technical point here. When you do that, you actually run into a problem that I don't want to go into here because it's fairly technical, but there's a way around it that is in the paper. And what you do now is you incorporate both of those sort of confounder coefficients, one related to the SNP data and one related to the gene expression data, into the mixed model. And when you do that, it does the right thing as far as we can tell from our experiments, which I'll tell you about right now very quickly. So we did some experiments on mouse data, and these are these same QQ plots. And what I've shown here is, as I said, nobody has really tackled this joint problem of population structure and the gene expression confounder problem. So what's shown here are models that tackle either one of them or the other, or some way that I've just created on the fly to combine them together because there were no existing methods. And what you'll see is that not a single one of them tracks the diagonal at all. Some of them are heavily deflated, which is bad. And many of them are heavily inflated. And the only one that actually looks at all reasonable is this model here, which is the model I just described to you. It's a linear mixed model that corrects for both of these things simultaneously. And when you use that model, you get a win on the number of hits compared to the next best model. Just for example, you get 4 times as many hits. And you can do a cis enrichment test, which relates a bit to what Gil was talking about. Basically, you can say of all the pairs of genes and SNPs, I expect pairs where the SNP is near the coding region of the gene to come out to have a stronger association, because those are just more likely biologically to be a hit. And these are called cis hits. And so you can do an enrichment over all your hypotheses and say, is my method finding cis hits more than it's finding non-cis hits? And then you can compare the different algorithms, say which algorithm is better at doing this type of cis enrichment. And when you do that, our model again wins across the board. And lastly, there's a lot of contention in the literature about this point, but many people argue that you get these artifacts, these so-called transbands, where basically if you plot the data in this way with SNPs on the horizontal axis and gene probes on the y-axis, that you get these lines here up and down, and that these couldn't possibly be real, and that if you have a method that makes them go away, that you're probably doing the right thing. Again, there's a sort of bit of a contentious issue, but we certainly observe that ourselves. These are all methods that don't correct for the gene expression confounder, and these are all methods that do, and you can see that those vertical lines go away. So that's all.
Well, that's it.
So, maybe I'm interested, but in the mouse data, if there was interesting signal, wouldn't it be the case that at the end you need to run away from the error bar?
You would, but it's a matter of proportion, right? So for one, in the paper itself, it's a random subset of data. And I mean, you think of how huge that hypothesis space is, you still expect, just as you would for a genome-wide association study, I mean, you can think of the fact that for each gene probe, you're doing a GWAS. And therefore, you expect the overall proportion to be very small and barely visible.
Right. So did you ever see data sets where that phenomenon is actually visible, or you don't even expect that?
Do we expect there to be more than what we observed? Is that what you're saying?
No. Do you expect data where, you know, the most extreme p-values would actually be able to creep up or I guess up above.
I mean, right, you do see that on some data sets, and it
You
just
do,
depends
right.
how strong and what the proportion is, so, right.
Gotcha.
Yeah.
Right, cool. Thanks.
The cohort, so the data, sure, he just asked what was the size of the cohort I used. And so in this paper, there's both mouse data and human data. I showed you just the mouse data, which contained 19 strains with about 10 repeats each. We also applied it to a human data set, which had, gosh, how many people did that have? It was all Caucasian, and I think it was around 600 people in that one, yeah.
Any more questions? No? Okay, thank you very much. Okay, so again, the second talk of this session is Jim Hogan from Queensland University of Technology, and he'll be talking about BioPatML.
Okay, thanks, Simon. And thanks, as usual, to the organisers. And today I'm going to talk a bit about a pattern description language recently ported to the .NET environment, partially to take advantage of the MBF-related services, and also a new Silverlight-based pattern editor, which we've written to go with the language. So I'm going to talk a bit about biological pattern search in very brief terms, discuss some of the motivation for this particular type of pattern description language. And the take-home message for that really is that it's intended primarily to allow the principled combination of pattern descriptions. Okay, and I spend a number of slides going through a bit of a motivating example to talk about the sorts of environments in which that can be pretty helpful, just concerning our own background in trying to discover motifs in bacteria. Then talk a bit about the BioPatML structure and features. I'll gloss over that relatively rapidly, just giving you some of the basics because it's a very complicated and extensive language, and I'll just give you some highlights before moving on to a demo. Okay, so as we've seen in various talks in this and other conferences, genomes have no shortage of patterns, but it's actually finding the ones that actually mean something, which is the more significant issue. And you can look at pattern specifications or pattern descriptions in two ways, I guess. You can look at them as a template for directed search, or you can look at them as some sort of documentation for those patterns that you've found which have actually got some hard-won significance. And so the idea of a language like BioPatML is really to do both of these things. So to act as a documentary record, I guess, of patterns that have been found and are actually specific and useful. In part so that they're nicely preserved and people understand the circumstances in which they've actually been discovered, and also as a mechanism to facilitate reuse. And so the idea is that if you're going to look for complicated combinations of patterns, instead of sort of building up the building blocks, so to speak, from scratch in their entirety, then perhaps you can actually take advantage of some of the work that's gone before in a very rapid and principled sort of way. So we understand, I guess, from our own bitter experience and other people's bitter experience that searching for small functional motifs is hard and is even regarded as futile by some people if you have sufficiently short motifs, which of course is pretty obvious if you're looking for statistical significance. But the saving grace, if you like, is that functional motifs often travel together. And so you're able, in some sense, to make use of the co-occurrence of these things to help to try and improve the signal that you're dealing with. And that leads to the idea that it might be nice if we can define some elemental components, and particularly those elemental components that have some significance in particular environments, and then combine and reuse those in the service of novel pattern search. So the motivation for this language initially was looking for transcription factor binding sites and promoters. So this is sort of a canonical example. You have a gene which is being transcribed this way. We have a transcription start site, a gene start site. And the basic idea is that the expression of this gene is regulated by the action of an enzyme called an RNA polymerase. And that RNA polymerase is always sort of enhanced by the addition of a specific or family-specific sigma factor, this protein at the back here, which binds to specific short sites on the DNA surface itself. And that the quality of that binding determines the degree of expression, or at least the probability of good expression. It's notoriously prone to high false positive rates, particularly in bacteria. It's not quite as bad in eukaryotes, but it's terrible in prokaryotes. And the traditional search in biology has been through the use of motif matches and position weight matrices, and more recently in other approaches like hidden Markov models and support vector machines. But taken by itself, using the short motifs, even in combination, you tend to find very low precision at any sensible level of recall. The reason that this then becomes interesting for a pattern description language comes when you actually have a closer look at the biology and understand that it's not quite as simple as the original diagram would suggest. I'm not going to go into all the gory detail of this. The point I really want to make is that the polymerase is a complicated enzyme of its own accord, but it also has additional complication through the various sites associated with the sigma factor, and as we will see, also through the action of additional proteins which tend to loosely bind to the enzyme complex. The main point that I want to make here is that the binding probability tends to vary enormously, and that these additional proteins are thought to have a significant action in ensuring some decent binding probability when the primary binding, if you like, is not as good as it might be. The nice comment down here, which is taken from a biology paper, it has long been known that domains 2 and 4 bind to the strongly conserved -10 and -35 boxes. Except, of course, when they don't, because they aren't, because the conservation of those is rather mythical, shall we say. So people have at various times over a number of years looked at the significance or otherwise of a number of additional motifs in the DNA. Okay, so we have this thing here at around -16. The -10 and the -35 elements are sort of the canonical hexamers that are associated with this problem. Here's an additional motif at -16, which is thought to be significant. And we have these proximal and distal up elements, which are very loosely bound to the complex, and that's further upstream of the TT-GACA element. Now, once again, I'm not really interested for the purposes of this talk in the significance of each of those elements. We've actually analysed that in a previous paper and found that some of the, optimism was misplaced. But nevertheless, it is a very, very good illustration of why some sort of combination of patterns, particularly if those patterns have been confirmed, can give us some sort of insight and some sort of additional leverage when tackling problems of this nature. So the desiderata that we had in coming up with this sort of language is support for elemental motif-based search and variations on motif-based search, simple probabilistic models, things like the position weight matrix and slightly more sophisticated models like that. Easy preservation, sharing and reuse. A rapid combination of elemental patterns to form structured patterns for more complicated search, particularly, once again, loading up things that have already been found to be useful rather than starting from scratch. That's the idea that the patterns that have been used successfully should be available for others in a very straightforward sort of way. And the key point in all of this is not that the patterns don't exist. The issue here is the effort that's involved in taking the patterns from the literature and encoding up a classifier or a search tool to actually search for those particular patterns. The idea here is that if you have a preservation of those in some sort of standard format with their associated parameters, and you can just dump it in a format using an easy-to-use editor, and you can then straight away use the parser and do a search, then obviously you can do a lot more exploration. And that's the whole point of the exercise. So BioPatternML is a unified pattern description language with some simple motif elements at the bottom end. Okay? So here's a straightforward idea. One of those standard consensus sequences that is discovered occasionally is TT-GACA. Okay? It's very commonly seen. We can specify that very straightforwardly in a motif. Okay? And the motif element we see here has the attributes of the underlying alphabet, which can be DNA or amino acids or RNA, and the specified motif. And so this is a very simple search specification for a motif, right? We can go a little bit further and we can document it, we can give it a name, and we can also have an associated threshold. So, and of course, for a motif search, that's a pretty generous sort of threshold. That means that you only have to get half the bases right, okay, at the elementary level. Where it starts to get a bit more interesting is in the way that you can take these elemental patterns and combine them in a structured way. So here we have the series pattern, and in the series pattern, you can have a hierarchy of underlying patterns. Okay, these can in turn be series or set patterns, which can in turn consist of elementary patterns, and you essentially create a pattern tree with the leaves ultimately being simple pattern descriptions of some kind, either motifs or gaps or some sort of alignment or anchors or whatever, okay, which help you to describe the search that you're trying to deal with. And so in this case, this is the canonical promoter description. I have a minus 35 element, a minus 10 element, and I have a gap of a particular number of bases in between. The other approach here is the idea that if we actually manage to store these patterns and make them available in some way, then people can look them up and reuse them. And that's one of the ideas that you'll see in the tool, where we'll actually load up a couple of previously stored patterns. Okay, BioPatternML itself, just to summarize, it covers DNA, RNA, and AA sequences, and all the RUPAC variations on the symbols. It allows a principled aggregation of pattern types, motifs and gaps and loops, hierarchical patterns, support for pattern libraries, integrated scoring of matches so that you can have a combination of scores of matches of the elemental components being combined in a weighted fashion over the hierarchy. The tool itself is not a learning machine in the sense that those weights are adjusted in an optimal fashion. Okay, that's an add-on that can be done, but that's something that we haven't done as yet. At the moment, it's a weighted combination with the weights preset, and it's also incorporates existing patterns such as prosite and regular expressions and so on. Some of the BioPatternML components that are supported, a catch-all pattern, a gap, composition of a region where we're actually looking at the base composition. Regular expressions and motifs are pretty self-explanatory. The position weight matrix, which is like a column-specific probability model. Sets and series, ways of combining patterns in hierarchies, and various others which are relatively obvious in most cases. Inverted repeat of a pattern match, constraints basically ensuring that the matches lie in a particular region, and alignment of pattern matches. Okay, so BioPatternML.net. Okay, so BioPatternML was created in our group a few years ago, and the initial tool was written by a fellow who wrote it in Java. That's Stefan, who came up with the lion's share of the language. And what we've done in this piece of work is to take the BioPatternML parser and search capabilities and ported them to the .NET framework, re-implemented them in C# and replaced the existing data structures with data structures based on the Microsoft Biology Foundation libraries. What we've done subsequently as well, and when I say we, I'm referring mainly to Sam, who's the first author on this presentation. He's then implemented an interactive editor in Silverlight 4, which allows the interactive creation and execution of these sorts of approaches. So you can load up patterns, you can generate patterns, you can construct them interactively, and then do searches and have a look at things. So we have the pattern library access, incremental visual editing, and parsing in search, which is something we'll see shortly. The main reason for going to the MBF libraries was really the fact that they had some very, very good parsing support. And they had more parsers than us and were likely to be building more stuff on top of it. So MBF now provides the core infrastructure for BioPatternML.net. But one of the things that we were wanting to do is to make this available in a browser-based experience so that people can just use it anywhere and rely on large pattern libraries that happen to exist on a server. And so one of the things which we've done in a partial fashion at least, is then to take the MBF libraries, which are created for the .NET runtime, and partially port them at least to the Silverlight runtime. So basically that's the state of the Silverlight MBF port, and that's something that will be of significant interest to Mike and company. But the basic point here is the ones which are sort of greyed out, they're the ones that have not really been, well, have not been included in the port at this stage, largely because of parallel extensions and a certain amount of complexity in some cases. But, you know, it wasn't on Lawrence's agenda at that stage to actually disambiguate, I guess, or refactor to deal with the dependency on the parallel extensions. But most of the functionality of the MBF library is then available in the browser. Okay, so what I'm going to do is just do some very basic motif searches using chlamydial genomes. I'm not going to come up with an optimal answer for these. I'm going to show you the variety of things that can be done. I'm going to use a model which has been developed in E. coli, as very commonly done. Chlamydia is an obligate intracellular parasite, as we often say in papers about it. It's basically a bacterium which causes a lot of problems, namely STDs and heart disease and a variety of other things. So it's of significant interest, but of course it's not a widely studied organism, so as is common in this area, people use the model that's been developed in Subtilis or E. coli and use that as a basis for search. So the key features of the tool are file parsing, reuse of confirmed pattern settings, and hierarchical pattern construction. And so we'll try and do some of that now. Okay, we will demonstrate this in Chrome for a variety of reasons. It just tends to show that Silverlight works rather well even in Chrome. So what I'm going to do to begin with is I'm going to grab a whole bunch of genomes and I'm going to drop them and I'm going to let them parse away, which isn't terribly sophisticated, but I've always found it rather cute that you can take all of those genome files and just drop them in there and they'll go through the MBF parsers and load them up. And we'll see that in a moment. While that's happening, we'll go to another instance of it where they've already been loaded. And what I'm going to do here is I'm going to take the promoter -35 element and I'm going to drop it in. That will give us an existing pattern. Okay? So that's coming from the server. So what you see, you have a simple motif description, and if we click on that motif, we see the parameters associated with the motif itself, with the pattern description. So we have the alphabet, we have the threshold. The impact is actually the weight in subsequent combination, and of course that would be something that we would optimize with learning algorithms on top of it, and that's the actual specification of the sequence TT-GACA. Okay? So very simply, we can go ahead and just to give you some idea, we've got about 20 full genomes there. It's actually not that bad to just go ahead and search, so I might actually just do that. And what you'll see is it will analyze and you see the progress bar. So you're doing a search for a small motif of about size 6 across about 20 full genomes, each of which is about 1 to 2 million base pairs. So, and this is happening in the browser, so it's probably worth noting that that's really relatively rapid for what it is. And in a moment, that will come up. At the right-hand side, while that's happening, you'll see that there's the pattern templates with the regional, the simple patterns, the structured patterns, and the special patterns, and we'll visit those in a moment. So what we see down the bottom is the results that have come up from the search. These are in situ, okay, so that's the matches that come from Chlamydophila abortus with the complete genome being the whole sequence. And that's the similarity score, the start and end position of the actual match, and the location of the matches highlighted in the display that we've got down the bottom here. Okay, and so if you click on one of these others, then it will highlight the relevant match in other parts of the genome and so on. And of course, if you move down, you'll see the matches from all the others. And of course, there are a huge number of matches taken over all of those genomes. So we can blow that away for the moment. But I'll just come back. So this thing here is long managed to parse. What I'll do here is just go straight to the idea of a structured pattern. And what we'll do is we'll go with a series. And there's a slight usability issue here with this mouse, so please bear with me if I take my time in actually loading up a couple of things, as precision is necessary. So what I'm actually going to do is construct that canonical promoter. So here I have, once again, I'm loading that from the server, which is why there's that slight delay. Lengthy delay, actually, but we'll see. So we now have that as a sub-pattern of the structured pattern. Over this side, I'm going to take a regional pattern, in this case the gap, and I'm going to come along and I'm going to place it on there again. And I'm then going to come down here and I'm going to grab another simple pattern, the motif, and once again I'll drop that there. And you see that I've managed now to create a series. I've now managed to create a series with a motif, a gap, and a motif. This is my pre-existing motif, the one that's been loaded up. What I can do in the middle here is specify the gap. So I'm going to make this, I'm going to give this a threshold of about 0.6. I'll make this about, typically the gap in sigma 70 promoters is in the vicinity of 13 to 21 base pairs. And we'll just give it standard weighting. And I'll go to the -10. I won't bother naming these things. The threshold, I will do something a little bit saner than that, make it about 0.7. The impact will remain at 1, and I'll make this one as tar-tat, or tat-tart, or whatever. Okay? And so on that basis, I can then go ahead and search. I can basically deselect some of these genomes to speed things up, which is probably not a bad idea, but it's still not that bad. We need a deselect button, evidently. And that ought to do for now. So the point, of course, is that I didn't actually set up the series combination. Okay, when I click on the series, I get the same sort of parameter window, and I can set a threshold at the series level for the weighted combination as well. So it's sort of an overall threshold. That's not going to worry us terribly much because we're not trying to optimize here. And we'll just let through some of the results as we can see. And so basically what you've now got is this compound pattern appearing at various stages. The obvious extensions at this stage that we've not done are to have some sort of differential highlighting of the component matches so that it's clear that we have the structure. And that's something which is an obvious extension, which we'll do shortly. The other thing which is an obvious extension, which I mentioned later, of course, is then utilising the MBF facilities for alignment to allow us to take all the matches that we've got, collect those, and very quickly align them, which, of course, is a very important thing to do in this sort of context. So the final sort of thing to do, I'm running a bit short of time, I think, so what I'll do at this stage, I'll just come back to this final one. And what you can do at this point, if you're particularly adventurous, is you can decide, well, what I will do is I shall add one of those distal up regions, okay? And so I can do that very straightforwardly. I can bring in a gap and drop it on. I can also bring in a motif, and I can drop that on, except that they're in the wrong place. So what I need to do at that stage then is to come along and very carefully insert there, and very carefully take this gap and insert it there. And what you see now is a slightly different structure. I now still have the series pattern, but I have a new motif, which I haven't defined yet. I have a new gap, which I haven't defined yet, and the existing compound structure. And so what I'm able to do there with this motif, I happen to know its structure very well. It's A, R, N, R. Okay, I just happen to have that with me. And I can actually then come in with the same sorts of parameters for that search. And the gap I would insert, and I would make, this is a relatively trivial gap, so I'll just put it in place with a very straightforward sort of set of parameters and the usual associated weights. And at that point, I'm now in a position to go ahead and hopefully repeat my search. And I will now have a search which is incorporating the other elements, okay, so which enables me to get a more specific result, because of course one of the things which plagues this problem, even when you use the compound structure, is that you've got a tremendous number of false positives within the ranges that we're talking about. And so the addition of these co-occurring elements is one way of reducing the false positive rate, and indeed probably the only way of substantially reducing the false positive rate. Okay, so that'll come up in a moment, and we'll see the compound structure. Okay, so on this occasion you see that we have a very, very close match at the start there. So we have really essentially one error against the specification that it was given for that distal up element, and the rest of the pattern match is similar to what we had before. So that gives you a quick overview of some of the capabilities of the tool. Sorry, I just trying to come back to a sane sort of thing. So the future of this, better support for sharing and publication. At the moment, we don't have any export facilities, and that's an obvious thing that we need to do. The alignment of the result elements and the coloured highlighting of component results. We have used this specification in another published paper where we actually did some sort of incremental search to optimise the weight parameters, but we haven't integrated that with the pattern description language and the tool properly. And so that's an extension which we will clearly want to do. The other thing is that we've previously published support vector machine work using K-mer-based string kernels. And the fact that there's support for K-mers in the MBF libraries makes that another thing which is very attractive for us. So definition of kernels for SVM approaches and expectation maximization approaches along the same lines as meme are the sorts of things that we would see as the obvious sort of learning support on top of this for weight optimization. So to conclude, acknowledge external research for support for some of this work. The MBF team, Mike Zukowski and Simon Mercer, various biological collaborators. And returning to the first one, I would tend to highlight Sam Toe, who's done most of the programming for this and is another very good student to come through our group. Thank you.
Hey, are there any questions?
So I'm not going to ask about Silverlight, even though I want to, and I'm so excited. I think that's awesome. The one thing I noticed was you said you were running the search algorithm in the browser, and I wondered if you've thought about doing the search processing on the server side. I don't know if the data's too big to send up. If that doesn't make sense, maybe the speed's okay as it is, but just wondered if that was something you guys considered.
Well, the previous version of it before the MBF port, the MBF Silverlight port was undertaken by Lawrence, actually relied on a web service. So the parser was actually sitting on the server, and the search was accomplished on the server. I think really what we want to do with this in the longer term is really to give the user the option of doing it locally, because see, for the student-based or the lab-based exploratory work where you have a very limited number of sequences, the sort of stuff that people might do and might sort of play around with visually in Excel or something, then that's the sort of thing which I would envisage people just wanting to do locally, because they're kind of fresh sequences. They might not be full genomes, and that sort of processing makes a lot of sense locally. But sort of canonical whole NCBI world database searches don't make a lot of sense in the browser. So in that situation, I would imagine that the server-side processing is a very, very sensible thing. But it would be good if we can give them the option.
Thanks.
Any more? No? Well, thank you very much.
Okay, thank you.